Okay, so let's look at an example here. So this is a worksheet you've been assigned. We'll get to the estimating size and estimating size in a moment. So let's just go to the calculating magnification because that's what we've been talking about. Okay, so drawing size, measured size, same idea here. Actual size, is the actual size of the organism. So let's look at like kind of this first one as an example. Okay, so we want to figure out what the magnification is of this. Now, here's the problem. We're in millimeters here, and we're in centimeters there. So what we need to do is we need to convert the one to the other. So I always convert to the biggest number. It just makes, um, you can make the calculations basically a little bit, well, no, sorry, I always convert the biggest number to the smaller number. So we want to do 2 centimeters. We want to know what that is in millimeters, okay? So what we do know is 2 centimeters. We know that in 1 centimeter, there are 10 millimeters, okay? So if those cancel out, we're left with 20 millimeters, okay? So that's for our drawing size, or our actual size here. So what we could do here is we could put that is equal to, sorry, that's not it. We know that this is equal to 20 millimeters. Okay, so now we can figure out the magnification here. So we do the drawing size, which is 20 millimeters. We divide it by the actual size, 0.5 millimeters. And that gets us our total magnification of 40x. Okay, so the trick here, it's a pretty easy calculation, right? The trick here is making sure that the two units match. Okay, so I'll let you do the rest, but that's an example of one. Um, Now the other thing, and again, if we were in school, we would do this, is estimating size and calculating magnification. Okay, so this particular question, right? We're looking at algae from a pond. Okay, um, now this is saying under high power. It shows 4 cells here. Okay, so this is high power. Now, from your microscope lab, we didn't do that lab, so I put it in the table below. So high power has a field of view of 0, has a field of view. So our field of view is 0.5. Okay. So it wants to know approximately well, one, it wants to know the diameter here, and that's 0.5 millimeters, okay? And again, that just came from down here, no calculation required, sorry, millimeters. And then the second one, it wants to know approximately how long is each cell. So we're basically saying how long is that, okay? So again, pretty easy, 0.5 millimeters divided by 4. How did I get 4? Well, there's 1, 2, 3, roughly 4 across, okay? So then that'll equal, each one is approximately 1.125 millimeters. Now, we could also convert that into micrometers if we wanted to. A lot of times cells size are expressed in micrometers. So we know that 1 micrometer equals 1,000, sorry, 1 millimeter equals 1,000 micrometers. So we could also say that it's 125 micrometers across, okay? So that's just a really quick, rough way to estimate cell size. So question 2 here walks you through a couple other examples that I would like you to try out, okay? And then the calculations down here I would also like you to give those a try. Okay? All right, thank you.